All right, everyone, welcome to day 8 of Unit 3. Today we're going to be discussing our intro to enzymes, and we're going to take some notes on enzymes. If you want to download those notes, the PDF notes for that, they're going to be found in today's agenda for day 3.8. What we're also going to be doing is finishing up our macromolecule stations. Hopefully you were able to finish that last class period, but if you were not able to complete that, you're going to have some time after the notes today to complete those. And just a reminder that that assignment, the macromolecule station assignment, that's going to be due at the beginning of next class period. So again, today's agenda, we're going to kind of mix it up a little bit. We're going to switch 2 and 3. We're going to do the enzyme notes first and then do the macromolecule stations at the end of class if there's time. So we want to make sure that we have all the information for the enzymes first. So without further ado, let's go ahead and jump into the notes for enzymes. We probably don't have a lot of chemistry background yet, but one thing that we can understand with chemistry is that every single chemical reaction that happens is either going to be producing heat or getting hotter, or absorbing heat, getting colder. But in chemistry and physics and thermodynamics and things like that, things aren't necessarily like cold. Like when you put ice or you put water in an ice tray and you put that in the freezer, you're not like putting cold onto the ice. What you're actually doing is removing the heat from the water. And when you remove the heat, then it'll turn into ice. So there's really not a thing that is cold. Like cold is not really a thing. Cold is actually just an absence of heat. Just like how there's no such thing as darkness. There's only the absence of light, right? So like a shadow is not something that you can generate. It's like you don't have a flashlight that can make a shadow. You have a flashlight that can make a light, and then you can block that light with certain objects and have the absence of light. And so cold and heat is kind of like that, where the cold is basically the absence of heat. Okay, so that's the first point. We've got that down, that all chemical reactions either are going to be producing heat or absorbing heat. All right, once we know that, we can kind of separate those two different reactions into their categories. The first category is going to be called exergonic reactions. Exergonic reactions release energy. Most of the time, that energy is going to be in the form of heat. So there's like a subset of exergonic reactions called exothermic reactions, and therm referring to like temperature, so heat. But exergonic reactions release energy. An example of an exergonic reaction, the internal combustion engine of a car, when it burns gasoline, that's going to be something that not only produces heat, because like engines get hot, but it also produces motion. So those are both different types of energy. Wood burning in a fire produces heat. Cellular respiration, so that's the process of like you eating food, that food getting broken down and then turned into different forms of energy. So like we use that energy for movement, we use that energy to keep ourselves warm, things like that. Those are all examples of exergonic reactions. And the key thing I want to kind of make a note of is this graph over here. This is really important to understand the difference in energy levels between the reactants and the products for exergonic reactions. So over here on the x-axis, this is the course of reaction. This is basically just like time moving linearly. Just we're going to have this straight shot of time, and then as the reaction goes on. And then over here on the y-axis is the free energy, or like how much energy the things in the reaction have. So the reactants are going to be the thing in the very beginning. So like before you actually do the reaction, that's going to be called the reactants. They're going to be at a higher energy state than the products or the stuff at the end of the reaction. And so you can actually see that energy was released throughout this process. So for example, like the reactants in let's talk about this campfire. So the reactant would be oxygen gas and obviously the wood, like the logs in the campfire. So the wood itself is going to be at a higher energy state. You can burn wood more than you could burn the products of that. So like the product of a campfire after you've burnt all the wood is going to be like ashes, right? Ashes are not going to be very useful. They're not going to have a lot of energy. You can't burn the ashes to keep you warm. So the amount of energy is going to be much lower at the end than it is in the beginning. Another example is going to be the hand warmers here. You know that if you've ever used one of these in the wintertime, you open the package, these things are going to start reacting with the oxygen in the air, and they will last a couple of hours. Over here it says about 7+ hours. So the chemicals in here are going to be reacting with the oxygen, and those reactants are going to be at a higher energy state. And as they turn into the product, they're going to be releasing energy, and that energy is again in the form of heat. So all of those examples are examples of exergonic reactions. The next type of reaction that we can see is something called an endergonic reaction. And these are going to be reactions that absorb or store energy, sometimes in the form of heat, sometimes in the form of a chemical or something like that. So an example is like an instant ice pack. If you've ever played a sport and someone on the team has an injury, you can use one of these instant ice packs. They last for a couple of hours maybe. Basically, there's two components inside of here. And there's like a bag inside of this outer bag. So once you break that bag on the inside, those two components can mix, and then it'll start absorbing energy. So those two reactants are going to be at a lower energy state, but once they mix, they're going to be at a higher energy state. And in order to get to that higher energy state, they have to absorb that energy from somewhere. Okay, and so they're going to be absorbing that energy in the form of heat. So these ice packs, they're not generating cold. So like going back a couple of slides, there's no such thing as cold. There's only an absence of heat. So they're not making things cold. They're only removing the heat from other things. So that's how these ice packs work. What about this diagram here? This is photosynthesis. Obviously, we've learned about photosynthesis maybe in middle school before. But basically how this works is that the plants are generating the final molecule here is going to be glucose, and glucose is a chemical that is used not only for fuel for the plant like food, but it's also used as a structure. Like it builds the stem and all the leaves out of that glucose. So that's the final product of the photosynthesis is glucose, but the reactants are going to be everything that the plant needs in order to make that glucose. And what do plants need? They need carbon dioxide. They need light. I know it says sunlight here, but it could be any form of light. And then water. So water comes in, carbon dioxide comes in, and then with the help of light, which is the main energy source, right? That's going to help drive this whole process of photosynthesis to make a product that is at a higher energy. And so endergonic reactions absorb or store energy. And of course, there's going to be a balance between exergonic and endergonic reactions in biology. And so we see both of those examples. Now, moving on to our topic for the day is enzymes. What is an enzyme? What is an enzyme? An enzyme is a type of protein that catalyzes or speeds up a reaction. So the word catalyze, that is a verb of something like a catalyst. And what is a catalyst? A catalyst is something that makes another thing happen. It kind of is it's like a tool that basically helps you do a reaction faster. And so enzymes are these tools that exist inside of cells that help reactions occur faster. They are not a reactant and they're not a product. They're basically a tool that is recycled after the reaction so they can be used again and again. Okay, so they're not used up. That's the key thing to remember here on this slide. Enzymes are not used up. They're tools that can be used. So similarly to like if you're driving nails in with a hammer, hammers are not a one-time use. You can use a hammer thousands upon thousands of times, and it can drive many nails, and it can do that same thing, that same task over and over again. Enzymes can do that same thing. They can do it same task over and over again. And enzymes work by lowering the activation energy. Okay, so what does this mean? We've got a great chart here to kind of explain this whole process, and this is kind of tricky to understand at first. But let's talk about this red line here on this graph first, and that's the activation energy. We have reactants and we have products over here, and the reactants are at a higher energy state than the products. So that's going to be an exergonic reaction, right? Reactants that are a higher energy state than the products. But the difference between this graph and the one that we see over here is that there's a giant hill in the way, right? And so what is that hill? Well, let's think about, for example, something that is at a high energy state than the products. And the example that I'm going to say is like gasoline. You can have like a cup full of gasoline or like a flammable liquid. And by itself on a counter, it doesn't really pose a huge danger, right? Gasoline we know is flammable. It's extremely flammable. But by itself on a counter, it's not going to do anything. It's not going to just combust on its own. if you want to turn that gasoline into its products of, carbon dioxide and whatever comes out the exhaust of a car, that's going to be the products down here. That's going to be at a lower energy state. What do you have to do to that gasoline to get it to that point, to burn that gasoline? Well, you have to give it a little bit of energy. Inside of a car, that's going to be a little spark from the spark plug. But if you just had like a little bit of gasoline on a surface, you could apply a flame to it, right? That flame is going to have a certain amount of energy, and that's the amount of activation energy. So once we get to that point, we've activated the reactants by heating it up to a point where it will get to overcome this hill, then the reaction will occur. So the activation energy is the energy needed to start the reaction. Okay, so going from reactants to products, we have to overcome this hill. We have to get the reactants to a specific energy state. Now, what do enzymes do with all of this? Enzymes help lower the amount of energy required to get to that product. So basically lowers this hill in order to get to the products. It does not change how much energy the reactants have, and it does not change how much energy the products have. That's really important to understand. The amount of energy between the reactants and the products is the same. The only difference is with an enzyme, it makes that job just a little bit easier to do. So it lowers the hill and makes that reaction easier. Enzymes make chemical reactions in the cell happen faster than they would on their own. And in many cases, these reactions would not happen on their own. So it's really important to have enzymes in order to overcome this activation energy so that we can get to the products. Okay, and so here's like an example of processes, like to kind of make it more tangible and relatable to real life scenarios. Like here are two processes. If you have without an enzyme, we are chopping vegetables. It can take a really long time. It's a process that, you have one tool that is helping you chop the onion here, but it's not really going fast. But if you have a specific tool that can help you do a process faster, like this thing, it's called a slap chop. That's the enzyme that is going to help make that process go much faster. Okay, so it's lowering the amount of energy required. So the difference between going from a full vegetable to its slices, in this case, requires more energy, and that's that activation energy. Without an enzyme, it requires more energy to get to the products. In our case, the products in this example are going to be chopped onions. Over here, we're chopping some vegetables, but with this help of this enzyme, it lowers the amount of energy needed to get to that final result, which is, chopped vegetables. All right. What I would like for you to do now is, or you can do this at the end of class, it doesn't matter, at the end of this video. You can watch this video on YouTube. There's a link to it on the agenda, but also in the notes posted in the day 3.8 lesson agenda. I'm not going to play it here just because I don't want to have a copyright strike on YouTube and have this video taken down. So you can watch that video on your own, but it's the function of enzymes, substrate, active site, and activation energy. So I would actually recommend you pause right now. You can come back to this video later. You can watch this video. It's just under 3 minutes, so you can take a little break and watch this video now. All right, if you haven't already, you can take a little brain break, about 3 minutes or so, but we're going to keep going in this video. Talking more about enzymes, we want to understand that enzymes only work with specific reactants or molecules. So I've been comparing enzymes to like a specific tool that you might have in like your garage, or maybe like in your kitchen, you have a specific utensil that you want to use. Enzymes only work based off of their shape. Like enzymes have a shape and they can only do one specific thing. So for example, the enzyme lipase breaks down lipids. Protease breaks down proteins. Sucrase breaks down sucrose. Lactase breaks down lactose. And so we notice that there's like a little pattern going on here with these examples. What pattern do you notice in the names of the enzymes? Well, if you haven't noticed yet, they all end in ase, right? So that's going to be true for a lot of proteins. Or a lot of enzymes rather, they're going to end in ase. And that kind of also describes what the job is. So like if you see sucrase, you can already understand that's an enzyme that breaks down sucrose. Lactase is an enzyme that breaks down lactose. So it's kind of really easy to understand what an enzyme does based off of its name. All right, so based off of that video that we saw earlier, that YouTube video, that 3-minute short clip, there were some terms that we saw in there like substrate and active site, and we're going to talk about those right now. A substrate, that's going to be the reactants that the enzyme acts on. So every single enzyme has a specific shape and it has a specific job that it can do. So the substrate is going to be the thing that it's going to work on, right? So in this example, we have this gray enzyme, and that enzyme has a specific shape where it has like this weird divot here and then like what looks like to be a 90-degree angle almost right there. And that specific shape aligns with this specific substrate. So this enzyme can only work on this substrate because it has that shape. And that shape where the substrate comes in, that is called the active site. The active site is the portion of the enzyme that attaches to the substrate. So in this example, in this diagram, we have the enzyme, and the active site is right here. The substrate is going to come into the active site. The enzyme will actually change its shape very slightly so that it better fits the substrate. And then we form what's called the enzyme substrate complex. All right, so this whole idea, you can see like there's a little bit of a change from this part right here to like now it's more of a true semicircle. This is called an induced fit. You don't really need to know that, but an induced fit basically means that it's kind of giving that substrate a hug. And then it forms this enzyme substrate complex. And then the enzyme's going to do its work. It's going to break apart that substrate into 2 separate parts. And then once it's done, it will release those products out into the cell or outside of the cell. It's going to basically release those products to do their job wherever they need to go. All right. And each enzyme works with a specific substrate. We say that substrates and enzymes are like a lock and key, right? So you can have multiple keys that look similar, but not all keys work on the same doors or the same locks. So for example, if you are eating a cheeseburger for dinner, amylase will only react with the starch found in the bun. It will not break down the other types of food molecules. So only one substrate lock per enzyme. So you have, if you're eating a cheeseburger, you have an enzyme that will break down the bun. You'll have an enzyme that will break down the meat of the hamburger or the cheeseburger. And then you'll also have an enzyme that will break down the cheese of the cheeseburger. And then whatever toppings or whatever things you might have in there, each enzyme will break down a different part of that food. So the active site for the enzyme is the specific area of the enzyme where it bonds with the substrate. And that's kind of like the lock and key. So you can kind of see like here are the teeth of the keys, and then they're the corresponding parts of the lock that fit that. So in this case, let's take a look at this diagram. What happens to the enzyme? We have this portion of an enzyme where we have an active site. Here comes a substrate. The substrate comes in, and then bonds are kind of weakened inside of the substrate after the substrate binds to the enzyme. And then finally, the products are then released from the enzyme. All right. We're going to do a little activity now in class, and hopefully it's going to be pretty easy to complete and hopefully not too messy. But what we're going to do is take 1 saltine cracker per person, and then everyone's going to take 1 saltine, and then, so every person in a group or a table will take 1 saltine per person. And then for the group, you'll also have 1 saltine cracker to kind of share amongst the 3 or 4 of you, how many of you are in a group. And then you're going to add about 20 drops of water to the cracker, and make sure you're doing this on top of a paper towel. The other cracker that's in your mouth, you want to make sure that you're not chewing it. You just basically, it's kind of gross, but you want to have your spit soak into the cracker itself. So just hold it on your tongue, and then you want to set a timer on your iPad. So maybe pull that out first. Set a timer on your iPad for 1 minute. And then after 1 minute, you want to kind of just compare. Don't spit the cracker back out. Just keep it in your mouth. You can swallow it at this point. You want to compare which one is soggier, the one in your mouth or the one with the water on it. So kind of compare those two, and then think about that. What does the water not have that your spit has? So I'll give you a hint. It's something that we've been talking about this entire lecture, and that's going to be a specific enzyme And so the enzyme amylase that we have seen, that's present in saliva, and that's going to be responsible for breaking down the cracker.